Number one, we're going to weave the evening through the three pillars of the Canadian Rare Disease Network. And so session one is about diagnostic advances. And for many rare disease patients and families, the journey begins and too often stalls with a search for answers, much like Ashley shared with us. Delayed or misdiagnoses remain one of the most painful and frustrating parts of the rare disease experience. Our first session explores how innovations in diagnostics, from AI to 3D facial imaging and real-world registry data, are transforming the diagnostic journey. And so I would like to introduce first, to represent Think Rare and the idea of harnessing AI for early identification of rare genetic diseases, Alexandre White-Brown. He's a clinical genetic counselor with the prenatal diagnosis team at the Children's Hospital of Eastern Ontario. He also holds the position of research associate at the CHIO Research Institute, focusing on the use of novel technologies and electronic medical records for the identification and diagnosis of rare genetic diseases. Alexandre additionally volunteers on the medical advisory board for the DLG4 Shine Foundation, helping to build a nonprofit organization to advocate for individuals and families with DLG4 related synaptopathies. Alexandre. And we're queued up.
Perfect. Thank you so much. And thank you so much for the kind introduction. And I really just want to start off by thanking you, Ashley, because I know that sharing a powerful story like that can't be easy. And I think it's important for us as researchers and clinicians to take a step back and remember why we do what we do. So thank you for sharing that. And thank you for making me think about why I put the time into doing what I do. And I'm really excited today, not to talk about the successes that Think Rare and our project has had, but more so to focus on the clinical impact that our project has had on rare disease patients and rare disease families at our institution in Ottawa at CHIO. So something that all of us here are probably very familiar with is that rare diseases themselves are quite unique and rare, but collectively they're quite common, affecting 1 in 50 Canadians. Now, for those 1 in 50 Canadians, many of those individuals remain undiagnosed to date. And for many who have already been diagnosed, they've endured a very long diagnostic odyssey, that referring to the time at which symptoms begin to the time at which they receive a diagnosis. And you can see on the left of the screen here in those green circles, there's many, many reasons as to why rare disease patients endure such a long diagnostic odyssey. Our team at CHIO back in 2019, 2020, we really wanted to focus on that one in the middle, minimal physician training on rare disease. And in 2019, the World Health Organization came out with a recommendation to equip frontline providers with tools for diagnosis and referral for rare disease patients to genetics to improve diagnostic processes. And building off of that recommendation, we want to explore a rule-based expert system. So basically what we like to call baby AI, basically an algorithm that uses rules such as if this, then that. Very, very simple. And we wanted to see if creating something like this could help us to identify undiagnosed rare disease patients in their electronic medical health record. And so the first thing that we needed to do is really put together a team. I know nothing about AI. 5 years ago. I know a little bit more now, but we needed data experts, we needed biostatisticians, and we needed clinical experts in this field. So we gathered clinical genetic counselors as well as geneticists with decades of experience in rare disease. And then we also found some individuals who work with our data analyzing and biostatisticians, and we put together our team. Once we had our team together, we really had to answer those 2 questions in the purple box or in the purple cloud. What items in a patient's chart do we want to flag, and can we even find these? So what we did is we went back and we looked at patients that were seen at CHIO between 2018 and 2021, specifically patients that received whole exome sequencing, a test that is funded by the Ministry of Health in Ontario and is specialized for patients who are suspected to have a rare genetic disease. We reviewed the charts of about 800 patients who were suspected to have this test and looked through to see what common data points they have, what commonalities do these patients have that might help us to build an algorithm for these data points and identify these patients so they can be referred to genetics and receive the testing that they need to get a clear and correct diagnosis. So you can see here that there's many different data points that we found when looking at these patients. Multi-system involvement, so many organs that are being affected in different parts of the body, and patients that are being seen by multiple different departments. So what we did is we put together all those different data points and we came across our first version of the algorithm. And now we weren't too naive. We knew that it probably wasn't going to work right away perfectly. So we need to do a very cyclical process to evaluate how this algorithm was going to work. So what we did is we had our data set, which was a cohort of patients seen from CHIO between 2018 and 2024, with about 370,000 active patients. And we ran the algorithm on all of these patients to see which of those patients will be flagged based on the data points that we had used. And our output was pretty high at the beginning, but we did these large chart reviews to try and find out, are we finding the right patients, or are we kind of finding some patients that might not be appropriate selection, so maybe don't have rare genetic disease. And when we did those chart reviews, we tried to find common things that might be explaining their symptoms that might not be a genetic disease. And we would make modifications and feedback to the algorithm. And we did this over about 1, 2, 3 years, over and over again, until we came to our final version, which was what we call now version 5. So as you can see on the right here, we have our exclusion criteria. So we know that there's patients at CHIO that we don't want to identify, patients who may already have a diagnosis, patients who have been seen and already evaluated in genetics, and patients who aren't within our catchment area, maybe over the age of 18. And then once patients were excluded based on that criteria, we have our inclusion criteria. And this is things like the diagnosis codes in their charts, so perhaps patients that have developmental delay and seizures, and then the departments that they've been seen in, so being seen in 2 or more departments such as cardiology or neurology. Once we ran our final version of the algorithm, we had about 450 patients that were selected. We had a large chart review of all 450 patients to identify which ones were appropriate selections that may have a rare genetic disease. And we sent letters to all of their doctors, all of their most responsible providers, MRPs, asking them to refer their patient if they feel that it will be appropriate for them to be sent to genetics. We always left this to the hands of the most responsible provider because they know that patient best, and we know that our algorithm wasn't perfect. There's many different reasons that a patient may present with symptoms that could mimic a rare genetic disease, such as prematurity or a congenital infection. For patients that were referred to our clinic, they were seen in our rapid access genomics clinic within 30 to 60 days of referral, and they were evaluated by a geneticist, Dr. Sarah Sawyer, and myself. And if patients met criteria for testing based on evaluation, we would offer that testing for them. And we're really proud that to date, with this project, we've provided 12 new diagnoses for rare disease patients who previously went undiagnosed. And our diagnostic rate is roughly 75%. So that means that we're capturing the right patients. And when we have these patients referred, 75% of them are getting a diagnosis that they previously didn't have. And one of our favorite examples of this is this really long pathway. This is a real example of a real life diagnostic odyssey of patient A. When he came to our attention and was identified by ThinkCare, he was 17 years old. His diagnostic odyssey actually started before he was even born. He was identified to have a ventricular septal defect, a congenital heart defect, prenatally, and would end up going through a 17-year diagnostic odyssey, being seen by 9 different outpatient specialties across 150 outpatient visits. Our ThinkCare algorithm would identify this patient at 17 years of age, and he would be evaluated by Dr. Sawyer and myself in our rare disease clinic. We would offer trio whole exome sequencing, which would identify a de novo variant in CHD4 consistent with the diagnosis of Syfram-Hicks-Weiss syndrome, which perfectly fit his clinical presentation and essentially ended this patient's diagnostic odyssey. Now, it was great that we had a diagnosis for this patient, but it was really the quote from his mother, who was in the room when we gave the diagnosis, that sticks with me to this day. She said, "It seems this would have been good information to have after he was born. Why weren't we referred to you then?" And I really didn't have a good answer for this mother at this time because it's, what can you say at that time? I really wish that we could have been able to provide this information to her earlier. But the next thing that she said to me is that now you can use this for other patients. Now you can start to end their journey sooner. And for her to say that in that moment was very, very powerful. You'll see in this word cloud here that this is just a subset of some of the diagnoses that we've made over the past year for patients identified by our thinker algorithm: Soto syndrome, Chung-Jansen syndrome, Birt-Hogg-Dubé syndrome. And for those clinicians and scientists out there, you may recognize that there's actually cancer screening recommendations directly for Birt-Hogg-Dubé syndrome. So for this patient who was now getting into adulthood, had never been previously diagnosed, we were actually able to start the recommended renal cancer screening for this patient. Potentially, saving his life. And some of you may have seen in the media or CHIO posts about some of the families that have been identified by ThinkRare and diagnosed. I really like to put their pictures up there with the permission of the families because they were really so interested in telling their story. And I think it's so important for us to look at these faces and see what this means to these families. Up in the top right, we have Eddie. Eddie is a 2-year-old male who presented with some differences in his development and a history of a couple of seizures. He was recently diagnosed with CHAR syndrome, and his parents really wanted to tell their story because having that diagnosis for them that early, it allowed them to have an explanation for what already happened, but it also allowed them to have a better understanding about the prognosis and the outcome and what to expect in the future for their child. In the bottom right, you'll see Anthony. Anthony is a 9-year-old, and he presented to our clinic along with his 2 brothers and both of his parents. Wonderful, wonderful family. And we actually, when we did testing for Anthony, we found a diagnosis for him, his 2 brothers, and his mother. And one of the quotes you'll see here was actually from a video that from an interview that this family did. And their father said, there's now less questions, there's less stress, there's less blaming ourselves. This opens up the doors for accessing resources and for us to understand what all of this really means. And they tell a very touching story about how their son, the diagnosis was given in English, but their son speaks French, and I was doing a little bit of translation, and their son started to cry after he finally received information and said to his father that this is why I am the way that I am. And in that video, if you want to go and search it on YouTube, the father ends up crying. It's very touching. So over the past couple of years, we've really done this retrospectively, so back all the way to 2018 to 2024, so that we make sure that we're not missing any patients that were previously not seen. Over the past year, we've really been focusing on identifying patients prospectively on a day-to-day basis. We've actually been able to run this as a clinical or as a silent best practice advisory study, which essentially means that patients are being identified in real time. They let us know through kind of a little alert, and then we can send the letters directly to the providers right away to have those patients referred. Through this silent alert study, we've actually diagnosed a patient as young as 3 months old. Right now, we're actually running a clinical best practice advisory, which means that as a patient becomes eligible or becomes flagged, it actually flags and sends a pop-up to the providers when they open that patient's chart. So every provider that's involved in that patient's care gets a pop-up, and they can make a referral by pressing one little tiny button, and that patient gets referred to our genetics team and gets seen within 90 days of being referred. Most importantly, and what we're doing in parallel to running our clinical best practice advisory is working on national and international experience. We don't want this to just be something that helps individuals in the Ottawa catchment area. We want this to be something that helps rare disease patients within Canada and around the world. And we're already working with Alberta Health Services, Sick Kids in Toronto, and McMaster Children's Hospital as well, those institutions that have the same electronic medical record as us, to get this implemented as quickly as we can. Then we're going to start to work on institutions that maybe don't have the same electronic medical record as us. And lastly, we want to see if we can do this internationally as well. We have a lot of work going on in the background to try and make this as accessible at no cost to everybody and all the institutions that we can, as we know that this will help to improve rare disease diagnosis. And lastly, and one of the most important slides, is really our team. It takes a really large team to put something like this together. Dr. Kim Boycott is here, and you'll hear from her later this week if you're attending the CORD conference. But every single name on this slide has put in a significant amount of hours to make this possible. And all of those diagnoses that I mentioned before are thanks to all of those names on this slide. And I'll leave it now to any questions that anybody might have. Thank you so much for listening.
going to maneuver a couple of things here. So I'm going to check online quick. If anyone has a burning question in the audience, I'll give them the floor right away and see if anything's popped up online. So I think in the interest of time too, we give you a break from grilling questions and keeping things to time, my main job. Thank you so much. So for those folks in person, of course, we'll be breaking a little bit later this evening and we'll get a chance to break off. And if anyone has a question that pops up for Alexandre, I'm sure they can confront him and connect and ask that hot question for sure. So as we move along, we want to explore 3D facial imaging for rare disease diagnosis. It's a project that Dr. Benedikt Hallgrimsson is a part of. He's an internationally recognized biological anthropologist and evolutionary biologist whose pioneering research integrates developmental genetics, bioinformatics, 3D imaging, and morphometrics to study phenotypic variation. Dr. Hallgrimsson leads a research program that explores the mechanisms driving anatomical variations, structural birth defects such as craniofacial abnormalities, and the connections between evolution and development. Welcome.
Thank you very much. Thank you for the invitation. And sorry, how do I do this? Oh, okay, great. And thank you, Jordan, for taking time out of your studying to do such a great job of emceeing this tonight. As Jordan said, I'm going to talk about facial imaging and the diagnosis of rare disease. I'm going to start with an image.
Nova Scotia.
Sorry, I actually thought I was showing a picture, but I guess I'll start with a short video. I meant to show a picture, but I guess we're showing the video.
Nova Scotia is somebody I would love to date. Nova Scotia is
commercial.
Something to go back
Before
to.
the video.
Here he is now.
Hi, I'm just trying to make sense of
another
all this
commercial.
data.
Just get Sasvaya.
Sasvaya. No!
Can we skip that somehow? Okay, let's just skip it. Okay, I actually had meant to show you an image of a young man with Louis-Dietz syndrome. His name is Tyler, and his family had graciously allowed me to use the video, which clearly didn't work here. It's an inspiring story. I would recommend you go and see it if you can. If I had shown it, then many of the medical geneticists in this room would have immediately recognized the face of, or would immediately recognize that the disease that Tyler had is Louis-Dietz syndrome. And that's kind of a remarkable thing. Why is it that the face tells you so much about what might be going on inside the body? The reasons for this, I think, are threefold. One is that the face is incredibly polygenic. So there's a huge number of genes that determine variation in the shape of the face. How polygenic, we actually don't know. It's for sure thousands of genes. We know that it's at least as polygenic as stature. Stature may be associated with the entire genome if you have large enough sample sizes. So this may also be true of the face, but this creates an enormous potential for a gene that is associated with a disease somewhere else in the body to also be associated with variation in the shape of the face. And that leads to the second reason, which is a phenomenon called pleiotropy, which is familiar to the geneticists in this room, but this is the tendency for a gene that does something in one part of the body to do something else in a different part of the body. So the combination of these two things, enormous polygenicity or genetic complexity, along with pleiotropy, creates a huge potential for a disease that may be a metabolic disease or connective tissue disease to also be associated with some effect on the shape of the face. The other thing about the face, though, that is distinctive and maybe is different from, say, stature, is that facial shape is a highly complex trait. So it can vary in all kinds of different directions. Everybody in this room has facial shape variation that is distinctive. We're varying in different directions from each other. Think about stature. Stature, you're either just shorter or taller, right? So it's a one-dimensional trait. Facial shape is a high-dimensional trait. You can vary in all kinds of different directions. So this creates the potential for the pleiotropic genetic effects that are associated with the disease, but also with facial shape, to also be associated with some distinctive direction in multivariate space, which is potentially diagnostic. So this is why facial shape is potentially useful. So these three reasons led us about, I guess, 12, 13 years ago now to pursue this project whereby we would enroll people with known genetic diseases and quantify them in 3 dimensions, and then see if we could use the 3-dimensional shape of the face to diagnose genetic disease. And I had numerous partners in this. François Bernier, who was here, was one of the main ones, as well as Mike Innes is over here. And we, over the years, amassed, with the help of all of these generous participants, a database of something like 7,000 people with known genetic diseases. And then we used advanced AI techniques and all kinds of multivariate statistical methods to see if we could use this information to diagnose genetic disease. And it turns out that when you look at the variation that you find in this sample, you see that there are very significant facial shape effects associated with numerous genetic diseases. Each point on these graphs is not; these are not individuals; these are mean effects associated with genetic diseases. And to put them in context, the orange spheres here would represent the amount of variation that you would have among, say, superpopulations of humans. And you can see then that the mean effects associated with genetic disease tend to be large. They tend to be larger, say, than the differences that you see among populations of humans, which overall account for about 15% or so of total variation in facial shape. So how does this actually perform in terms of diagnosing genetic disease? Well, it turns out that we can, in a sample of people with about 112 different genetic diseases, we can diagnose the correct disease with about 80% accuracy. Which is remarkable, because that's you're just doing this from the 3D image of the face. This was a remarkable enough finding that the Francis Collins, who was the director of NIH at the time, he stepped on a couple of years ago. This is during the height of COVID, and we believe that this was his first non-COVID related tweet in 2020. So he was distracted enough by our study to tweet about it, and he thought that this was a finding of significance. Which, of course, Francis Collins, if you know anything about him, he's a very well-known geneticist. So one question that's relevant to this, actually to the image that I would have showed you at the beginning if I had, is what is the nature of this variation that's associated with genetic disease? What does it look like? Does this variation go off in new directions, or does it tend to follow directions that are already present in the background population? And if so, that actually leads to some interesting applications of this work that I'll talk about. So it turns out then that the facial shape effects associated with genetic disease tend to fall along extremes of normal variation that you see in the background population. This is just a statistical decomposition of the facial shape effects of genetic diseases. And the thing I want you to notice is that if you look at these distributions here, these are syndromic means. And you can see that there are these elevations on the ends. So these are specific genetic diseases that tend to fall along these axes of variation that characterize the background population. And how does this happen? It happens due to this phenomenon called morphological integration. This is the tendency for traits to co-vary with each other due to some underlying developmental factors. So you can see this, for example, in achondroplasia. This is, of course, the well-known actor Peter Dinklage, and he has achondroplasia, as is well known. And he has the facial features associated with achondroplasia, which results in this very deep set expressive eyes that he uses to great effect as an actor. But he also has a highly flexed cranial base, as well as the frontal bossing of the forehead. And the degree to which he shows these features is going to be related to the severity of his underlying disease. It's related to the degree to which the growth of cartilage in his development has been perturbed. And so this is the phenomenon of morphological integration. This suggests then that, and we know, for example, that that cartilage development, this feature that is affected in achondroplasia, is also something that drives normal variation in the background for the population at large. And so this then leads us to think about, could we take these syndromic vectors and project them into the background population and map them for genetic disease? I think I'm getting way too complicated now for most people in the audience. What's that? And so if we take these vectors, these disease-associated vectors, and map them and look for genes, we can say something about the relationship between the disease phenotype and variation in the background population. And so we've done this for achondroplasia as well as a number of different diseases, and this is what the result looks like for achondroplasia. And you find all kinds of genes that in fact are related mechanistically to cartilage development, to the underlying mechanism of achondroplasia. In other words, this disease specific axis is associated with background variation in processes that are related to that disease. We then used a method in mice where we could take the genes that we found for this syndrome axis in humans, and we used the method that we developed in the lab called multivariate genotype-phenotype mapping, where we look at the combined effects of all of these different genes. And we asked the question of what phenotypic effect do they produce? And if you've ever looked at mice with achondroplasia, which I guess is probably a small proportion of this audience, you would know that the animation that I'm showing you here basically replicates what mice with achondroplasia looks like. And this is a remarkable thing because we've replicated achondroplasia in mice without the causative mutation, which is in a gene called fibroblast growth factor receptor 3. And so we've taken this axis of variation that's associated with achondroplasia, taken the genes that are associated with that axis, asked the question of what do these genes do to mice, and we replicated achondroplasia, at least the craniofacial effect. So this suggests then a model for thinking about the genetics of rare disease in larger context, in the context of background variation, where we can think of these diseases as extreme versions of variation that's present in the background population, that is present among all of us here. And this is important because it may relate to the ability to use the severity of things like facial shape variation to predict underlying disease severity. Now, does that actually work? I can't tell you for sure that it works for many diseases. It's going to depend on the mechanisms of the particular disease. But we have started to look at this in connective tissue disorders, and actually the diseases like the one that Tyler has, the one in the video that I was going to show you at the beginning. And we found that the genes that we associated with variation in these Louis-Dietz syndrome axis are also associated with the genetics of non-syndromic aortopathies. And people with Louis-Dietz syndrome, they tend to die from aortic dissection, from aortopathies. And so this then gives us a plausible, or at least a plausible hypothesis then, that there is a relationship between the severity of the facial shape phenotype and the severity of the underlying disease phenotype. In other words, the severity of the facial shape phenotype may be clinically useful. And this is a direction that we're currently pursuing. Here's some circumstantial evidence that points towards this probably being the case. If we compare people who are relatives of people who have genetic diseases, particularly parents, we find that they tend to be further along the disease axes for the disease of their affected child. And that's kind of remarkable. That indicates that it's not just the, sorry, it's amazing, yeah. It's not just the causative mutation, it's also underlying background variation. And this suggests then that individuals, particularly those who get diagnosed early and have severe disease, they may have genetic backgrounds that are also associated with more severe disease. In other words, the model might look something like this, where you have individual, say, who has subclinical diseases, proband A, maybe the relatives of that individual are really far from the threshold for the disease on this integrated axis of underlying variation. whereas an individual who has more clinically severe disease would have relatives that are closer to the threshold. This is different from a standard threshold model in the sense that the axis here is this integrated axis of variation that is connected mechanistically to the disease. It's these syndromic axes of variation that have mechanistic connections to the disease. Okay, so I have three conclusions from this. I hope I wasn't too complicated for this audience. First of all, facial shape is potentially diagnostic because there are many pathogenic variants that are also associated with variation in the face. So we can potentially use 3D facial imaging as adjunct tool for diagnosis, and there are multiple applications for that. But secondly, this works because variation in facial shape is high dimensional, but also highly structured. And this structure is something that we can leverage. We can potentially use the structure of variation to use facial shape or other phenotypic traits that are associated with a disease as measures of underlying disease severity. And that may be a clinically useful feature that might actually help kids like Tyler, who I was going to show you the image of at the beginning of the talk. So thank you very much. I hope I stayed to time, Jordan. Yeah. Okay. Thank you.
No, I let you off the hook. Are there any questions in the room for Dr. Hallgrimsson?
I mean, I spoke too fast.
Over there.
Yeah.
I'll let you ask it and I'll repeat it.
Mm-hmm. Yeah.
I'm curious if you can get into
Where was that? Oh, okay, great. Yeah. Mhm. Yeah, yeah.
if you do understand this, but understand what causes a de novo genetic mutation that's not linked to the parents. Is there any way that we'll understand like where that comes from?
Can I ask that question for online crowd?
Yeah.
So the question, if I can summarize it, is how do you, for de novo genetic mutations that aren't expressive present with the parents, how do they then present with the child?
So it's a great question. I mean, so mutations occur randomly. So we all have mutations, and occasionally those mutations are associated with a disease. There are people in this audience better positioned to answer this question than I am because they actually deal with this exact issue all the time. The implication of the work that I'm talking about, though, for this particular question is that it may explain why some individuals who have severe disease and thus get diagnosed are different from those who have a less severe disease and maybe don't get diagnosed. And then this may be occurring because the individuals with the more disease, with the more severe disease, that de novo mutation is occurring against the genetic background where they're already shifted towards the direction of having that disease by the same underlying mechanisms. So if you take achondroplasia, for example, I mean, it's a bad example because it's a highly penetrant mutation, but you can imagine that that an individual who has a de novo mutation for that, but born to parents who have a relatively short stature with short limbs and facial features that are more like achondroplasia, they're going to have a more severe phenotype than individuals who are born to people who look like Jordan, right?
Talk about stature. Can I ask you quickly, as someone who's absolutely terrified that in 2 short years of residency, I'll find myself in a position with a panel of patients in family medicine, what are the challenges from a clinical perspective for helping someone like myself in getting from where you are now to where this might be out there in the world for people like me?
Well, I think again, there are also people in the audience that can answer this question as well or better than I can. The biggest hurdles actually are in making this technology work in such a way that it actually aids clinical practice. So you've now seen this. You see how busy clinics are, how difficult it is to work something into the workflow of a clinic. So that's that's a big barrier. It's not enough to show that a technology is potentially useful. You also have to be able to show that it works within a clinical setting to make the end product of that clinical encounter more useful and more productive. So there are multiple steps there. I mean, Francois is probably better at answering that than okay, yes, that's a great one. Yeah, so what we are working on making the imaging technology easier. Right now, we use expensive 3D cameras. They take space in the clinic, so that's a problem. We have really accommodating collaborators at the Alberta Children's Hospital, so we're able to do it here, but that may not work everywhere. But we are working on an app that will work on your phone. And once that happens, you could do this anywhere, including in Uganda. Okay.
Dr. Hallgrimsson, thank you very much. Well, our final speaker of session 1 is going to explore patient registries with us. We've in some way explored that in harvesting data and where we can find it most useful and where we can implement it in some of these research projects. I want to invite Dr. Lawrence Korngut to the stage. He's a neuromuscular neurologist, researcher, and entrepreneur dedicated to improving patient outcomes through clinical research, innovation, and data-driven healthcare solutions. As a professor at the University of Calgary's Cummings School of Medicine and a member of the Hotchkiss Brain Institute, he specializes in diagnosing and treating neuromuscular conditions, performing electromyographic studies, and advancing real-world health data collection to drive better care for patients with diseases like ALS. And the last time I spoke with Dr. Korngut was on a research podcast that I led just before I started medical school. It's great to welcome you to the stage.
Thank you, and congratulations, Jordan. It's a blink of an eye. At least it felt that way to me. I suspect it was longer for you. So let me. Okay, so I'm Lawrence Korngut. I'm a neurologist here, as Jordan mentioned, at the Neuromuscular Clinic in Calgary. So I see adult patients with diseases of muscles and nerves, many of which are genetic, many of which are acquired inflammatory autoimmune conditions. And our community has really been blessed by this surge of new therapies for different diseases. And of course, looking back over the last 15 years, when I was starting out my career here in Calgary, I don't think we really foresaw exactly what was going to happen and all of the challenges that would come along with that, and really the need to organize ourselves as a community, as you're doing here, of course, as you've done here. And what I hope to share today is just a little bit of kind of what we did and what we're excited about and where we're going in the different things that we do. And I think it's important to recognize that there are many problems, there are many pain points to address. And so often there are many solutions coming from many different perspectives. And I think that's part of what I get excited about. One of the diseases that's near and dear to my heart is facioscapulohumeral muscular dystrophy, which you may be familiar with, FSHD. We have about 20 biotechnology companies steaming towards the first treatment. So it is an untreatable muscular dystrophy at this point. But certainly as we think about what happened in Duchenne, spinal muscular atrophy, and other diseases, we're sort of laying the groundwork for what do we do if and when we have a positive phase 3 trial and we need to deliver this therapeutic to our patients in an efficient way, make sure it's covered, make sure everybody who needs access has access. And so these are all challenges we all struggle with across our respective conditions of interest. But I'll just share a little bit about where we've been, what we're doing, and we'll go from there. So, always important to show disclosures. Much of the work that we've done, my research team here is based in Calgary, but there's collaborators and really essential team members across the country. But much of our work has been funded by academic grants, but probably the majority has been funded by industry partners. And so always really important to recognize. So our spinal muscular atrophy work, for example, for the last decade or so has been funded by Roche, Biogen, Novartis. et cetera. And so we of course have rigorous protocols to ensure that our data is as objective as possible, et cetera. But certainly that's important. And the second part of my talk that I'll briefly highlight today is a company we started about 7 years ago called Lumio, that spun out of our research lab. And I'll touch on kind of what we're doing there and what we're excited about from that perspective on a global level. So the first thing to highlight is just as in every community, the Canadian Neuromuscular Disease Registry that we started 15 years ago, there's I think 136 investigators at this point. And so this is a large group of people. We have very clear governance. We have lots of documented procedures for things. And so importantly, while I get the privilege of sort of speaking about it, that's where the work is really done at all the centers across Canada. And of course, our team members, some of whom are going to be at the conference over the weekend. And so what this looks like as a registry is certainly we're active in all the neuromuscular clinics across Canada. So this is 36 adult and pediatric neuromuscular programs, 14 ALS clinics, as I mentioned, about 130 some odd investigators. And so virtually we're in most clinics. Now, we don't have capacity to collect data on every single neuromuscular disease. There's about 140 different conditions that are eligible to enroll in our registry. But we do focus on specific ones that I'll highlight, where we then are able to focus, generate data, run programs. But interestingly, there's great value in terms of the diseases we don't even collect data in, where we just know who has the condition and where they are and which clinic they attend, when we plan for clinical trials, when we try to evolve the work that we're doing. And so I think there's a lot of different benefits to this kind of work. And the other thing I'll mention about this registry is when we started it 15 years ago, the neuromuscular community was quite fragmented. Researchers in one part of the country didn't really know the researchers in the other part of the country. Nobody really understood who was doing what. And certainly this led to us getting organized. So while I'll talk about the data, the story is really about community. It's really about clinicians coming together, meeting researchers, meeting patients, meeting the advocacy groups, the charities within our space, Muscular Dystrophy Canada, ALS Canada, Defeat Duchenne Canada, Families of SMA Canada, Cure SMA Canada now. And really that's been the story, I think, of our neuromuscular community sort of pulling together around this, which we kind of did just in time as we got hit with all these therapies, which we were very excited about. But then it was a matter of like, okay, well now what? Because now we have patients who don't have access that we feel should, and how do we advocate for all of that? And so here's a list of the different conditions that we follow. And as you'll see, we're kind of actually speeding up our work at this point in terms of adding new diseases. We just launched our FSHD data set across Canada. Our collecting data will be announcing the big funding that came through in the near future. And so we follow about 6,000 patients across Canada, across these different neuromuscular conditions. And then this is going to be a little bit small, but here you basically see it's a quite a large number of partners that have come together to do this work. And so you'll see the universities across Canada. You'll see groups like the Center for Disease Control in the US. Canadian Drug Agency was one of our partners, and in the last 2 years, I would say we've been really working closely together in terms of demonstration projects of how do we do our job collecting data, and how do they do their job using it for useful purposes, because it really takes everybody to kind of participate in that process. You'll see the industry partners, and then of course, indispensably, our patient advocacy organizations that really got the ball rolling for us, have always been there to support us. And that's really been tremendous and important. My favorite statistic isn't how many of us there are or how many patients we have, but it's how much data have we actually used for something interesting, useful. Have we had some impact? So we've had about 330 data uses at this point. Many of these quite involved projects, some of these smaller data inquiries trying to plan for clinical trials, et cetera. You probably can't read, you probably don't want to, but you probably can't read all of these different sort of outputs in terms of the researchers. And what's interesting is I've led this sort of registry network for 15 years now, but probably about 80% of the publications don't even include me, which is wonderful. That there's so many researchers, so many scientists or industry partners doing work and leveraging that data. So spinal muscular atrophy is a great example. A number of drugs get approved. A lot of questions come up immediately. What does this patient population look like? How much does an infant with spinal muscular atrophy weigh? What is that growth curve like for planning of the dosing of that community? So sometimes it's really quite simple, obvious things that we just don't always have the information for or in one place to obtain. So providing data to regulators was something we were really excited about, because in the end, we just want this to be useful for the community. Same thing for network advocacy. So advocating to payers or advocating to the Canadian agency, this was CADTH back then, so Canadian Drug Agency now, in terms of how should we track these outcomes once a therapy is available. Back in those days, we were thinking, how do we prepare for clinical trials? We were so excited about preparing for clinical trials, we forgot to prepare to actually, how do we get the treatment to the individuals once the trial is positive? It just almost seemed a bit unimaginable at that point. I think we're doing better now in terms of being ahead of that curve. But then how do we advocate as a network? So let's say there's a new clinic that needs to be launched in Saskatoon because Saskatchewan does not have any coverage for a given disease. All the clinics across the country can write a letter saying, this is what we have, this is what we expect as the minimum standard of running a multidisciplinary clinic for this disease. We can provide that to the neurologist. We can provide all the data from across the country, all the data from Saskatchewan, and then they get resources to advocate. I mean, if it was only that easy, but that's a great case example of how the ALS clinic was set up in Saskatoon. And then in terms of working together, so much of this is how do we come together as a community? When we look at what's happening in FSHD now, these 20 therapies moving through the pipeline of development, hopefully at least one of them will make it into the clinic. That takes everybody. It takes, of course, volunteers, patients, families, takes clinicians, clinical trialists, takes industry, takes all of those different components. And so having a network around this is really important. So we did set up a network which is now operating as NMD4C, which is our national neuromuscular network run by Hans Lockmuller from Ottawa, and doing a brilliant job. But it is interesting how the coming together to kind of, we initially thought this was sort of a data thing, and it really became more of an opportunity to collaborate. And then data, of course, came out of it. And so that really has been my greatest learning. We participate in the Treat NMD Global Registry Network. So this is 37 countries. For SMA, for example, there's almost 10,000 patients participating in that. Just to put it in perspective, I think we have about 60 individuals with SMA in Calgary, probably growing much faster now than in the past. And so as we work in FSHD, as an example, Project Mercury, which is based on the success of Project Hercules in Duchenne muscular dystrophy, has a network of 25 countries, basically preparing and laying the groundwork for when that first therapy hits. Probably, in the next 24 months, we're hoping there will be a positive phase 3 trial. And that's just incredible. I've never seen organization like this before. And when we go to meetings, it literally is everybody like in this room. We have clinicians, researchers, families, patients, industry partners, and then, we all know what's happening and we're all getting ready to do this work together. And maybe I'll skip over that. And so that's been the work led by our research program here from the University of Calgary, again, with a whole bunch of universities and brilliant researchers from across the country. And again, what we learned was there's a lot of value to coming together, working on these tough problems, just as you're here to do, collecting very rigorous real-world data to then advocate in a very credible, data-driven way. But we also learned along the way that you can really benefit individuals much more than just advocating for access to a drug. There's much more to life than just access to a therapy. Of course, this is something that's critical to have a treatment, a disease-modifying treatment for your illness, but there's much more than that. So 7 years ago, we actually spun out a company called Lumio that really focuses on really benefiting communities using data that can be collected. And so here's a picture of the Lumio team from just this past week. And And basically, we do the same work as I mentioned in the Canadian Neuromuscular Disease Registry, except we do this now in 18 countries across 14 different disease areas, ranging from common diseases like migraine, bone and joint disease, all the way through to rare disease, of course. And really, the concept of the company has been to enable us to develop software that allows us to advance this kind of work, and then selfishly to bring it back to Canada and to the neuromuscular community in a way that we were never able to accomplish on an academic level, because we did try. And so I'll just give one example. And so earlier this year, we launched a program called Better Life FSHD. So this is a patient engagement solution. And really what it does is it allows patients to track their own outcomes, understand what is happening in their community, connect to clinical trials, and have their data contributed to research. So in the US, so we have an FSHD registry in Canada that doesn't exist in the US. And so somebody mentioned earlier, how do we, as Canadians, help, of course, our own community, but then how do we share and impact others around the world? And there's certain things that I think would be difficult to compete with. We're not going to compete on AI computing capability with some of the big American, programs and companies. But we do have a universal healthcare system, and we're shockingly collaborative. So whenever I speak at meetings where we have neuromuscular registries from the US, what's interesting is we have sometimes quite comparable numbers of patients in our programs. But it's because we're very comprehensive in Canada, and in the US, it's all divided into little competitive groups. And so that's a big advantage. We're also much less expensive in general, probably about 30–40% less. And so what's interesting in the US, they don't have a national FSHD registry. There's a bunch of smaller ones, which of course do amazing work. But the FSHD Society in the United States wanted to have a one-stop shop solution for patients to engage with them as a community. And so we launched Better Life FSHD in the US. The first cut of this was about 530 patients. And what we found was really incredible uptake. 97% of patients reviewed it very positively. The amount of information. Now, this is an online registry, which is different than what we've been doing in Canada, which is a clinic-based registry where you're collecting, hospital captured data. But what's important is that they're different, and they kind of have different purposes and different advantages. And so what's interesting is we just received funding to launch Better Life in Canada for FSHD. So that's going to be rolling out in the next probably couple of months. And what's interesting about Better Life FSHD Canada is we're then able to take all the data collected. It's governed by the physicians across Canada that oversee the working group for the Canadian Neuromuscular Disease Registry, and all the data will essentially be held by the Canadian Neuromuscular Disease Registry. And so it'll be subject to the same sort of governance principles, open to the whole community for use. Anybody, I think, as I mentioned, we've done over 300 projects with the CNDR. What's more important is we've never said no. So if anybody's wanted data, we've always provided it. And so we're very excited about this. And then as a final point of what we're excited about is this Better Life solution got so much uptake. It was so positively received by the community in the United States. The FSHD Society has such a positive relationship with the community, but also with all those biotechnology partners who are developing all these treatments, that one of the major setbacks in that condition in FSHD in the past year was there was a phase 3 trial that everybody was cautiously optimistic for that turned out negative. And so this is, of course, a huge setback. We're all preparing to kind of how do we advocate for this drug to be administered for our community. But what ended up happening as perhaps a bit of a silver lining is the company then committed to providing all the clinical trial data from all of their programming up, all the phase 1, 2, 3 to that point, all of their tissue samples, everything that they have to the society. And so what we ended up doing is launching what we're calling Focus FSHD, which is an open platform that anybody can request and obtain not only this highly sensitive and very detailed clinical trial data, which we're going to make completely available to the whole community, but also there's going to be samples, biosamples, blood, muscle, et cetera, which is just incredibly exciting. This has been done previously in ALS to huge success. and probably shaved off 3 to 4 years in some cases of these companies trying to develop a treatment for the disease. And so we're very excited about it. But anyhow, I thought I would just share those different aspects of the work that we're doing in the neuromuscular community. Again, 99% of what you've seen is not me. It's of course everybody across the country. We have 2 team members here, Brianne and Grace, if you guys have time to meet. And then, yeah, thank you.
Thank you, Dr. Korngut. There was a comment online from some of our folks joining from across the country of open science and wondering about that. So thanks for addressing it from your perspective in some sense. I think to state a time, we'll move ahead and we'll take our break. Now is the time for our break. And for folks who might have some questions online that are still burning, I'm monitoring that. Maybe as we get back in, we might be able to touch on some of those quickly. For those of you here in person, there are some snacks at the back and some drinks and a chance to mingle. We were originally scheduled to restart at 6:15. I think I'll push it to maybe like 6:20, 6:25. I'll give a 5-minute call at 6:20, and then at 6:25, we'll get everything started again so folks can mingle or take a break online. Some restart with session 2. I'm wondering if I can put Dr. Hallgrimsson on the spot for a minute, because there was a question that popped up online and we moved along. And so I've got like a minute to, and maybe I'll hand you the mic. So the question is for the facial recognition software, how is it taking into folks from, is it European based for now? What is the variety of folks from different ethnicities being incorporated into the data?
So that's a fantastic question. Unfortunately, the cohort is more European centric than we would like. And so we can't fully answer the question of how well it will perform for people across the globe. We have some indications of that, though, because there is diversity in the sample, and the test that we've done so far suggests that there isn't a big difference in performance. But we need a more diverse sample in order to be able to verify that. So it's a work in progress, and it's a challenging one. There are socioeconomic reasons why, particularly in North America, when you go to patient meetings, you tend not to have, say, a high representation of Black Americans. And there's also, for very good reasons, often suspicion of research in the healthcare system. Yeah, and so those are challenges that we are working on. And it's a fantastic question.
Thanks. So we will restart with session 2. And session 2 takes us to that second pillar of CNRD's mission, and that is from discovery to treatment in advancing rare disease therapies. And so, as many of you know, there are a few no approved treatments for many rare diseases. And here in Canada, access to clinical trials or innovative therapies can be inconsistent and often completely out of reach for many. And we've touched on that a little bit so far. In this next session, we'll dive into advancements in therapy development from promising treatments to transformative technology. And so I would first like to invite Dr. Peter Canoe to the stage to talk about moving from discovery to treatment in advancing rare disease therapies. Dr. Canoe is a pediatrician and clinical geneticist specializing in diseases affecting the skin and bones. His research focuses on understanding biologic pathways that influence skeletal growth and maintenance with the goal of improving human skeletal health. At the University of Alberta, he leads efforts to identify novel disease-causing genes in inherited bone and joint disorders. His work includes studying osteofibrous dysplasia, primordial dwarfism, and scoliosis genomics. Dr. Canoe.
Thank you very much for that kind introduction. And thank you to the organizers for the opportunity to come today from Edmonton to talk to you all about rare disease therapies. So I work on two different areas in genetics. Just trying to work this out. So in my laboratory, I study the genomics and the pathomechanisms of rare bone disorders. But in the clinic, I'm also very interested in the advances and the therapeutic trials that are coming for the rare diseases. And this has all, really become available because of the Orphan Diseases Drug Act in America, maybe 20, 30 years ago, which really paved the way for investment into rare disease therapies and what we're seeing right now. So, when I started in genetics, for one, our ability to make a diagnosis was probably less than 10%. At that time, chromosomal arrays hadn't even come in. We were using chromosomes and then subtelomeric fish. And lo and behold, we've seen the ability of our ability to make a diagnosis reach about 40 or perhaps even 50%. And we're becoming more and more like mainstream medicine. And the way we practice genetics is also changing. And this has ushered in this whole era of precision medicine, right? So you have a treatment, but you need the right treatment for the right individual, because we know that some people may not respond to therapy X, and some people may do better on therapy Y. And that really ushers in the whole idea of pharmacogenomics in terms of what the body does for drugs. So in the space of rare diseases, we're not so fortunate to have, multiple drug therapies, but we do have a few that are already licensed, which I'd like to talk to you about today, and many more that are coming. So if you look at if you look at therapies for rare diseases, you think about, all these very rare conditions that present with short stature. Now, most of them are incredibly rare. They're probably about over 300, and the nosology keeps increasing the number of subclassifications. But if you look at them collectively, just like most other orphan diseases, they do make up a considerable number of patient visits to the clinic. And the nice thing about the skeletal dysplasias is that you can make a diagnosis on the X-rays. So it's very similar to how people look at the face and look for distinctive features to make a particular syndromal diagnosis. The 3D face imaging that we're talking about, many of the skeletal dysplasias can be classified using the X-rays. And so in some instances, genetic testing is just there as an icing on the cake because it allows you it informs reproductive risk counseling. It's also important because some people want to know what they have. But most of the times, the diagnosis may be secure just based on what we see clinically and what we find on the X-rays. So one of the most common conditions that causes short stature is achondroplasia. Many of you would have heard about this condition before.